Hello, everyone. Welcome to today's webinar, Optimize Your Multi-omics Research, Data Management Challenges and Best Practices. I am Karin. I'm the VP Marketing at Ontoforce, and I'm the host of today's webinar. So this webinar is organized by Ontoforce, and we're very pleased that ZS wanted to collaborate with us and share their insights and findings on multi-omics data. We have a packed agenda for the next 45 minutes. So let me already go to the next slide and introduce the speakers to you. So our experts for today's webinar are two people from ZS Associates, Rasmus Borup Hansen and Mark Österlund. And they're joined by my colleague Berenice Wolbrecht, who's currently VP Solution Enablement at Ontoforce. A quick introduction into the experts. So Rasmus and Mark are joining us from Copenhagen, the ZS office over there. Rasmus has a background in mathematics and computer science and has been supporting scientists from the IT side for more than 25 years. And for the last 11 years, he's been working with bioinformatics and different types of omics data, both in a commercial setting and public-private partnerships. And he will also introduce this in a moment in more detail. Mark, as part of Rasmus's group in Copenhagen, has an education in bioinformatics, systems biology, and biotechnology. He started out in bacterial and human genomics and public health, and has now moved into multi-omics in the private sector. Then my colleague Berenice has a background as a bioinformatician and has worked as a bioinformatician as well as a scientific engineer for companies like BASF and Institut Pasteur in Lille, in Belgium and in France. And she's been with Ontoforce now for a couple of years, working currently as a VP of Solution Enablement. What will Rasmus, Berenice, and Mark cover today? So they will talk about the potential of multi-omics data, the challenges regarding omics data in today's biopharmaceutical research. They will also discuss how life sciences companies around the world overcome these challenges, and we'll also look at some tools that those companies use to go through or to plow through the vast amounts of omics data. We end with a Q&A. Feel free to already submit your questions in the Q&A chat in Zoom here. We will answer your questions at the end. If we don't have sufficient time, we will get back to the questions that we weren't able to answer during this live session. So everyone will get a response to his question, either it's during this webinar or it's in the days after this webinar. Before I hand the floor to Rasmus, a short introduction into Ontoforce, because quite some people are new to Ontoforce. We saw from the registrations. So Ontoforce is a global provider of a data and knowledge platform designed specifically for life sciences companies to unlock hidden insights from data. We've been around since 2011. Our product is called Discover, and today we have more than 10,000 users across the globe. They come from the big pharma companies, but also the smaller pharma and biotech organizations. They are using Discover. We ourselves have operations in Europe as well as in the US. And here you find a glimpse of the Discover platform. It's bringing together data sets, internal data, public licensed data, and through semantic technology and knowledge graph. The searches that scientific researchers perform in Discover are much more powerful because the data gets linked to one another, thanks to the semantic technology and the knowledge graph. Our customers are deploying Discover in various use cases across the entire drug development lifecycle. We see customers using it for early discovery, but also preclinical, clinical studies, registration, as well as commercialization. But today's webinar is going to focus on omics data and the challenges, specifically when it comes to managing and dealing with the different types of omics data. That's it for the welcome and the introduction. Rasmus, I'm happy to give the floor to you now.
So thank you very much for the nice introduction. I would like to start out with very briefly talk a little bit about who we are. and where we come from. I will promise you it will not be a large sales pitch, but I will talk a little bit about the company that we come from. So ZS is a consultancy company. It was founded around 40 years ago, and it has its roots in academia. We are working with analytics and data science and technology. This is mainly related to healthcare and pharma, but we also have clients in completely different sectors like airlines. I need to get my computer to work. Here it is. So as you can see from this slide, we are a global company with more than 14,000 people and a large number of clients. It all started out in Evanston, which is just outside Chicago in Illinois. And now the company has expanded to Europe, India in particular, other Asian countries, as well as South America. One of the areas that ZS is working in is research and development. And if we zoom in a bit further, then we get to biomedical research. This is where Mark and I are working. So we are primarily working with custom bioinformatics solutions and workflows, but sometimes we also contribute in the other areas that you see here on this slide. So Mark, my colleague, will now talk a little bit about what omics is and what you can get out of it. So I will step out and allow Mark to present his part.
Hello. So I'll be going through a bit of the science. So what is omics? Omics is the concept of taking a biological system and measuring everything of a given type of data. There are multiple different types of omics. I'll here be go through 3 of them that are particularly relevant to the rest of this webinar. First, we have genomics, which is the DNA, the sum of DNA in your cell. And genomics, i.e. the genome, sort of gives you the potential of your cell. Then we have transcriptomics, which is when a gene is being used, it's being transcribed into an RNA fragment. And the sum of these RNA fragments gives the direction that the cell is going in. Then we have the intactome, the intactomics, which is interactions between biological elements in the cell. Typically, we look at protein-protein interactions, and where the other 2 showed you the potential of the cell and where the cell is going, the intactome shows you the scaffold, the structure of how the cell interacts with itself. And one classical example of this would be pathways, biological pathways. Then why look at omics? Well, first, let's take a step back and consider the classic scientific theory, scientific method in which you develop a theory, then you formulate a hypothesis to test that theory, you find some data that can either confirm or reject your hypothesis, and then based on that, you have a better theory or have rejected your theory. With omics, because we can reasonably say we have captured the entire distribution, the entire variation in the system, we can flip this script. Here we can start out with our observations, with our data, analyze that data to find patterns. Then from these patterns, we can look and try to form hypotheses that can explain what we're seeing in the data. And then we can mature these hypotheses into theories, which means that this is a data-driven hypothesis generating approach. And because this methodology is unbiased, we can discover things that we didn't expect. One example of this is when people started doing bacterial metagenomics, which is you have multiple different organisms in your genome sample. We started seeing a bunch of genes that didn't really match anything we knew. It turns out that we were discovering a bunch of new bacteria that couldn't be cultured in the lab with conventional means. And this kind of story happens all over omics sampling. So in essence, through omics data sets, we're discovering more and more of nature every day, which is amazing. Now, the ZS way of looking at things, well, the ZS way of doing a project can be seen here. First, we start out with some kind of data, typically multiple data types from multiple different sources. Then we analyze each data type individually to convert it into individual data layers, evidence layers. And these would typically be significant scores for proteins, but it can really be anything that is machine parsable and comparable. I wouldn't recommend midichlorians, but to each their own. And then from that, we combine these evidence layer into a meta evidence layer, which is here shown as networks. Now, a network, of course, comes from, in this case, the interactome, but can also be from the knowledge graph that you'll hear about later. And the great thing about networks is that you put things into context. So we might see that the most significant individual protein sits alone in a hairball of insignificant proteins. So the context means it might not really be relevant. It might just be your data that has some artifact in it. Whereas you could also see some smaller regions like this one down here, where you have several proteins that are each kind of significant, but not too significant. But all of them are only connected to other significant networks, other significant proteins, and they form these subnetworks, these modules that might be worth looking into. In this case, even the insignificant protein might actually be relevant. And this process of finding areas with more significant proteins we call enrichment. Now, an example of an actual graph we made. This is, we call it a benchmark plot. It is essentially a ROC curve. And what you really need to know is that you want to get to y equals 100 as soon as possible. So what we see here, we plotted the transcriptomics analysis as alone as orange, the genomics analysis alone as teal, and then the combined transcriptomics, genomics, and interactomics analysis as black. And this trend that the individual omics analyses perform very well, definitely usable, but the combination of them gives something much, much greater than the sum of its part. We see a much higher enrichment of relevant proteins towards the top of our prioritized list, which also means that the novel targets on the list has a much higher confidence, which is, of course, great. So while that sinks in, we have a poll question for you, which should show up on the screen anytime now. Great, here we are.
So what Mark just talked about was a case where we first analyzed the different omics types individually, and then we tried to layer them on top of each other, and then that way analyze them together. So that could give us some additional insight, and now we see the results of the poll coming up. At least I see it. I hope that everyone sees it. So it's like 50-50. So some people look at individual omics types and some other people look at them together. That is actually not really surprising to me. Many people, they only have access to individual omics types. So of course, that sets a limit to what they can do. But if you have some more insight from having multiple data types, then you can get much further. It will also, of course, depend on what kind of cohorts you have, what kind of diseases you are looking into, what exactly makes sense. But I think that we can say without any doubt that more and more people are going for having multiple omics types, ideally at the very same time from the same patients. And in that way, they will be able to do some more advanced analysis and get some stronger results. So all this does not come for free. We have tried to divide some of the challenges that we see across the industry into these 4 categories that you see here on this slide. Many people, they face some simple practical challenges related to compute and storage systems. And that is because many of these systems that you need to analyze omics data are fairly specialized. Some people may try to use their own PC or an old high-performance computer system, but then you will very quickly run into performance or capacity issues because omics data, as you know, takes up quite a lot of space. So what we typically see that people are doing is that they move into the cloud, and then another challenge will be that they have to control how much money that costs, because if you don't really know what you're doing, then it can quickly accelerate in the cloud. In any case, there's also the aspect of omics analysis that it requires a lot of different tools that are specialized, and also, of course, the right skills to operate these tools. And that also brings us to one of the scientific challenges, is that these tools sometimes form different schools of how to analyze omics data. And these different schools, they take turns being the go-to solution for how to analyze a particular omics data type. So if you have to be on top and on the forefront of how to analyze your omics data, then you need to invest a lot of time on evaluating different tools and following where the field is going. If you look a bit on the business side, then the main challenge is, as we see it, that there is no single end-to-end solution that goes all the way from data capture to insight into your data. There are many tools that take care of some of the steps on that journey, but we also see that these tools can sometimes be quite expensive, and you need to invest a lot of time in them. So when they're not always working optimally together, then our clients very often have a hard time replacing them with other tools because they have invested a lot of time and money in them and built a lot of processes around them. So it's hard to take one out and put something in instead. Finally, the fairness, which I assume that most of you have heard of already, meaning that the data should be findable, accessible, interoperable, and reusable, is also an area where we see a lot of challenges with omics data. If we start with accessible, then a very, very simple thing that we see sometimes is that people give up getting access to the data even before they've tried, because they expect that there will be a lot of bureaucracy and procedures and boring forms to fill out. And they say, okay, this is too much work, let's do something else. So as an industry, I think we should strive to make sure that we have some very, very clear and simple requirements for getting access to the data that we generate. That could be a way that we collectively could address that issue. If we look at the three other aspects to fairness, then they have a thing in common that is called metadata. And that is a key to working with omics data. One thing that we see people struggling with is to simply be able to provide the metadata for the data that they generate. They know when they're conducting the experiments what it's all about, but writing it down or registering it in a formalized, systematic way in some system requires time and effort. And sometimes people say, okay, I will just put the metadata in the file names, then other people will figure out what it's all about. But that only works for a few weeks, maybe a few months for the same people. Then they would struggle to remember what they were actually working with after a while. We have also seen that's actually not that long ago. I was working with a client would not really want to put any metadata in the file names, because it turned out that they feared that they would be held accountable for the metadata, and they really just wanted to essentially register the readouts from the instruments and put them into the computer, and then someone else should struggle with all the metadata. So we often see a lot of pushback when we are talking to the people out there regarding providing metadata. And then if data should be truly findable, then there should also be a tool or a good environment for searching the metadata that you do register. And then the problem will typically be that there are many ways to register, say, a disease. There are different ontologies you can use. There's human disease ontology. You can use a ICD-10 code or a number of other things to register your metadata. And if you really want to have your data that's interoperable, then you actually need to make sure you capture all the metadata, not just which disease was examined, but also some other information about maybe the patients, everything you can identify. Otherwise, it's very hard to reuse your data in another setting later. But if we do get all this data, then you will be able to integrate data from different experiments, maybe even across cohorts if it's done in the right way. And then you will be able to do some kinds of analysis like Mark was talking about previously. And luckily, we have some very good friends at OntoForce who are very good at handling metadata. So I can see there is something in the chat. I will just see if it's a question. No, it's just asking you all to ask questions. So I will end my part here, and we can take questions later. So I will stop sharing.
Thank you, Mark and Rasmus.
Yeah, welcome. It was a pleasure.
I think the echo is resolved now. Yeah, good. Then you give me a really good handover. Then you end up with the fact that it's important to put metadata to be able to search for this information. Then I will share my screen. Okay, you should be able to see my screen. I will just hide this different part. Good. Then why is it important to have metadata to make them findable? And today, researchers, and as you mentioned, are spending about 20% of their time looking for information. It could be a question like, okay, do we have sequencing data for patients that have been onboarded in a certain clinical study and for which we have detected an adverse event? It could be like, do we have proteomics and metabolomics data for a certain disease and for a certain gene panel? And all this information, if they are not well tagged with meta information, then they really look like searching information in an haystack. Why? Because data are generated from different silos of information, from different ELN, from different source database that could be internal, external. And secondly, they don't necessarily share the common vocabulary. For example, there might be some experiment, some information that will be tagged with a disease with NSCLC. Some other might mention about the non-small cell lung cancer. Then if I'm not aware about this diversity in the data, I can miss out in some important information. Then I will explain how knowledge graph can support your journey in searching for this kind of information. Then basically, knowledge graph allow you to integrate various information all together. Then typically how OntoForce is dealing with omics data and knowledge graphs, then we are integrating omics metadata from different data sources that could be internal data or external data, together with ontology that will allow us to realign the data from different repositories. And for example, realigning the fact that disease might have been mentioned with different names or from different classification, but also with some omics results, for example, which are the genes that are differentially expressed in a certain experiment, and also eventually with different biological knowledge or again, internal or external. Could be that, for example, that a certain genes has been already tested as a target in your pipeline, or it could be public knowledge about like publication around a certain biomarker. Then how knowledge graph are supporting your user journey on omics research. Then knowledge graph will support different steps across the typical data lifecycle that is about searching for information, selecting the relevant data point, analyze the data, get into your findings, and store back your result. Then where knowledge graph will be supporting your journey. First, knowledge graph can help you to explore various resources across multi-omics landscape. Then the fact that they get all the metadata from different platforms, different resources are coming together, give you one environment where you can query, explore the data. Secondly, with the fact that the data can get reharmonized on the knowledge graph, you will be able to effectively select all the experiments and data of interest on your journey. When you have selected, for example, sample of interest, that will be analyzed on analytic platform. Out of the analysis, for example, a differential expression analysis, you might find differential expression, some gene candidate. This gene candidate, you want to explore what kind of knowledge is available for this gene. Then that is, again, a place where the knowledge graph will be able to support by bringing information about what is known about this specific gene. And again, your findings out of your final experiment will be again stored, typically in a data repository, a data warehouse, or a data lake. And again, you will research your journeys. And knowledge graph are very good support for exploring, select, and build your insight. I will shortly go into a demo to show you like how can look like an integrated experience on a knowledge graph. And for that, I will use a demo platform on Discover, for which that contain different omics resource all together into the platform. Then on this platform, there is a lot of different data sources that have been integrated, that contain different omics resources, that contain ontologies and knowledge information all together. And basically, that gives you a knowledge graph that I will show you as an overall. And basically, we will be able to explore this knowledge graph to find your search of interest. Then let's say that I would like to know which are the samples that will be available for me to analyze for a certain disease that I'm interested in, that is NSCLC. Then I'm typing NSCLC. The first thing is that the system will search like which are the data source, which are the is that will be available. Alors, excuse me, I'm just log out of the platform in the meantime. I will just relog in. Then I can search which information is available on the NSCLC. What Rasmus mentioned about the tagging of the information and the use of standard allow you to make sure that you will not miss out to information. And for example, here, I can extend my search, and NSCLC is in fact recognized as a synonyms of non-small cell lung carcinoma, and I can extend my search with the synonyms. You will see I get much more result when I extend the result. Now, I can go specifically to select some experiment or select some sample. I will go to sample. Then when I click here, I will have my result on the right. I will just hide my history. I can have result on the right. I can see, for example, is it something that is publicly available, or is it something for which I need to request a control access? What kind of data source are available? If I have metadata on my sample about the library, proteomics, genomics, etc., I will be able to filter down. I can make my selection of sample of interest here. And once I have selected my sample of interest, I can send this sample to my analytic platform. Now, let's say that out of my analysis, I come back with identifying one gene candidate, and that is EGFR. I can come back to the same environment to find out what is known about these genes. Then let me type EGFR, and I will go to gene because it's a gene that I'm looking for. again, I will just hide my history. I will filter to Homo sapiens as I'm interested into human. And here I have the EGFR gene of human. And with an integrated knowledge graph, I will be able, in fact, to reach out to all the information that is available for these genes. And you see that when I look for information about EGFR here, I'm able to reach out to information that are coming from very various data sources. And I can explore that. Of course, I can find the information about these different the different information about the gene, and I can continue to explore the knowledge graph. Then, for example, I can say, which are the cell line for which EGFR has been tested as a knockout, for example. And I will get here the list of cell lines that have been testing EGFR as a knockout. I can see it's associated cell line that for lung adenocarcinoma, it's exactly the disease that have been analyzed further. And then that will allow me to really explore the data and find supportive information to my search. We do have a poll question. Then the poll question is, what omics platform are you currently using? Is it an in-house built solution? Is it an external solution? A mix between the two? You don't have an omics platform, or you don't know which one you are actually using. Waiting a couple of more seconds for finishing the poll. Then about 50%, then you have about like average same between an in-house and an external solution. And 8% of you have a mix between an in-house and an external solution.
Then, then I will conclude this webinar here. So once more, Berenice, Rasmus, and Mark, thank you again. And everyone else, thank you for joining and for participating.